So, good evening to one and all present here. Rupali ma'am, few participants are saying that link is not working. Please check. In group, WhatsApp group, they have written. I have reshared the link. So I just received a message from Dr. Krishnani that he will be late by 10 to 15 minutes. Until that, we have assigned you work. Yogita ma'am, please share the link.
Yes, ma'am.
You please fill this form and a paragraph about a paragraph. Am I audible?
Yes, ma'am.
A paragraph about startup or any innovation which you want at your place. So it differs from one to another. दे रहा है सबको दे रहा है नहीं I am audible. Yogita ma'am, I am audible. Other participants, I am audible.
Yes,
Yes,
ma'am,
yes,
for
ma'am.
audible.
Okay, okay, okay. Thank you so much. Thank you so much.
I have shared the link in the group.
You share in the what in chat box also.
Yes, ma'am.
Yes. So all the participants who are present, they please fill this Google form. What type of discussed with another our resource person or with Nikul sir who can lead us to during and food processing. I have a good news to share. And for a start, So Assistant Professor Ms. Shurbi Srivastav and BSc third student Shailendri Mishra from the Department of Microbiology have procured fifth position in space technologies and applications for Earth and Beyond 2025 Ideathon for Viksit Bharat. The innovative idea was titled Aqua Guardian, Co-Creating a Viksit Bharat Through Water Innovation. And the second, which we congratulate Assistant Professor Ms. Yogita Lokhande and BSc third student Kajal Vaishnav, Department of Microbiology, for the esteemed achievement procuring second position in space technologies and applications for Earth and Beyond 2025 Ideathon for Viksit Bharat. Innovative idea titled Smart Climate Resilient Villages, a Tech Ecological Model. So congratulations to both of you.
Thank you, ma'am.
Welcome. Tomorrow, as we will be having valedictory session also, I'd like to inform you that there will be a test also. So all the participants, be ready. We will be sharing all the PPTs also. And the link for exam will be shared at that time only. Yesterday, the session, tomorrow the session will start from 2 p.m. First session, that is the session 11th, will be from 2 p.m. to 3:30 p.m. It will be taken by our expert, Mrs. Sujata Verma, Director and CTO of Argus Embedded Systems Private Limited, Hyderabad, on smart and sustainable farming. The second session or the 12th session will be from 3:30 to 5 p.m. It will be given by Technical Marketing Manager, Chaitanya Group of Industries, Malkapur, Maharashtra, India, by the expert Dr. Yogesh Narkhede. And the topic will be Innovative Agro Marketing. The third and the last session, 13th, will be on the topic Drone Technology for Precision Agriculture and Crop Monitoring. Expert for this session will be Mr. Gautam Kumar Haldar from Chhattisgarh, CEO Pikag Energy, Hatkoj, Bhilai, Chhattisgarh. And then there will be a test from 6:30 p.m. to 7:30 p.m. Online test and feedback. It will be simultaneously along with the lecture. 6:30 to 7:30, you have to complete your exam. online exam, and from 7:30 onwards, valedictory session will start. Yes, Khushbu ma'am, what question you want to ask? The hand. If any question you want to ask, you can unmute yourself and ask question. Speaker will be joining with us, so please cooperate with us.
Ma'am, good evening, ma'am.
Good evening.
Ma'am, test will be MCQ based?
Yes, MCQ based test will be on MCQ based.
Okay,
Yes,
ma'am. Thank you, ma'am. Yes.
okay, welcome.
Hello?
Yes, sir.
Ma'am, sorry, ma'am. Like any award for the marks will be there or the test what we write?
Yes. 80% marks you have to secure to get the certificate, sir.
Oh, okay. It will be based on the whatever the PPT presentations the scientists have given.
Whatever the PPT are being presented and what the FDP is having, that only questions will be there.
Okay. Thank you, ma'am.
Welcome, sir.
Good evening, ma'am. Hello. Hello.
Yes, sir.
What about current session?
Sir, can you repeat your question again?
Currently there is a session. No, anyone speak or just or guest has not arrived yet.
Sir will be join within some time.
Okay, okay, okay,
Within
okay.
10 minutes, sir will be joining because he was on a trip to West Bengal. Some
Okay,
director
okay.
meeting was there at ICAR there.
Yes,
So
yes.
he has reached at his place and just ready to join. Getting ready.
Okay, okay. Yes, yes, yes. Okay.
और सर वहाँ कैसा है सर?
बहुत बारिश है मैम.
बहुत बारिश है.
बहुत बारिश है. यूएसटीएम पूरा
डूब
क्या
गया.
बोलूं, मतलब दो साइड से रोड पूरा कट गया था पानी से.
ओ लैंडस्लाइडिंग.
हाँ, लैंडस्लाइडिंग.
लैंडस्लाइडिंग. ओके ओके.
जस्ट गेट का सामने वाला ही रोड.
अरे बाबा रे, बहुत पर आपका बहुत अच्छी जगह पे सिचुएटेड है सर. We enjoyed
तो
our
हाँ,
trip
आज
there.
पूरा एनएच पूरा जाम हो गया इसलिए, क्योंकि सबका गाड़ी, कोई एक भी गाड़ी पास नहीं किया.
अच्छा.
हाँ, और बैक साइड में भी जो रास्ता था उधर में भी लैंडस्लाइड होके पूरा मिट्टी भर के रास्ते में पूरा ब्लॉक.
बारिश में बहुत परेशानी होता है सर.
हाँ हाँ, बारिश में बहुत परेशान होता है.
ओके ओके. तो उसके
अच्छा भी
लिए
लगता है.
कुछ अच्छा, उसके लिए कुछ सोचिए सर, इनोवेटिव आइडिया कि उसको कटिंग को
नहीं
कैसे
वो
लैंडस्लाइड.
वो तो गवर्नमेंट फंड देने से हम लोग आइडिया दे देंगे.
आप आइडिया लाइए. गवर्नमेंट फंड अपने पास में सर है ना, निकुंज सर. ही विल बी प्रोसीडिंग अहेड.
श्योर श्योर.
Like this we can have few ideas, sir.
यस यस.
Now, Neeraj Sharma sir has asked that please explain about the test once again. Rupa Verma ma'am has also asked. We will be having MCQ test and few questions will be there. You have to secure 80% of the marks to avail the certificate from the AICT. And we will be sharing the link for that. And you have to go to the portal where you have entered your details. And from there only you will be filling your form exam. And it will be for 1 hour.
Yes,
So,
ma'am.
please explain in detail to all the.
Yes, ma'am. Actually, there will be a fixed time. है ना?
Okay,
In that
1
time
hour.
only you have to appear that test. That test will be from 6:30 to 7:30.
Yes.
Okay, so you all need to be punctual for that because there is no extra time given. Only 1 hour is limited by AICT. So in that 1 hour only you have to appear that test. Okay.
Tomorrow, ma'am.
Yes,
Yes,
tomorrow.
Okay,
It will
ma'am.
be MCQ and the questions will be from only the topics which are explained here. So the PPTs will also be shared with you so that you can prepare with those tests.
Okay, today you will share the PPTs.
Yeah. Today we will share the PPTs. Also, the questions will be from tomorrow's sessions also. So you need to be
Attend the
careful
sessions
listening
also.
attentive
Yeah.
for tomorrow's test.
Okay, ma'am. Thank you. Thank you.
Okay.
मैम, probable questions.
Yes, sir. Yes.
Now we are all student. So as per student, now we are asking need probable question.
Tomorrow afternoon you will be getting some questions, sir, that these might come.
Yes.
Okay.
So any feedback you want to give, that what improvement should we do for such programs, or what should be organized in the offline or online? They are very well. I request you your feedbacks over that.
Sanjukta ma'am today joined. First time I have seen her today.
Yes.
Good evening, ma'am.
Good evening, ma'am.
This is Geeta Ramani from KG College of Arts and Science, ma'am.
Yes.
Basically, I belongs to mathematical sector. Why can't we have a multi-collaborative sessions, ma'am? Through today till today, I need to know what and all implementation through the mathematics,
Okay.
but I didn't receive that one. Uh, Yesterday, somewhat on nanotechnology that will be comes under fluid dynamics portions, ma'am. So that is maybe some of the portions will be covered in that. But basically, you all know that nowadays maths not properly moving in all the college sectors. Students are not joining mathematics.
In
So
your area also. The
yeah,
same
ma'am. Yeah.
problem is with us also, ma'am, because some these new courses which have arrived, so the students are attracted towards that, and
Yes,
we are
ma'am.
not able to provide real what we say that the students just they don't want to read and have a degree. They want that they should be placed in a best position
Yes, ma'am.
for this, learnings. And
Yes,
unless and until we will create opportunities for that, we will not be able to attract the students in any field.
yes,
So
ma'am.
we can start and we can have that collaboration. And
Yes, ma'am.
please let me know that how maths can be attached with these fields.
Yeah, ma'am. I'm in the UBA team, ma'am, in my college.
Yes.
So that they asked to check the weather conditions in the tribal area using the differential equation like that, ma'am. It's a simple calculation only, ma'am. But we need coding also. Without computer science knowledge, we can't do it. But anyway, at least in our topic where we can apply that also we are not getting, ma'am. So only I'm asking, is there any type of collaborative learning? Lot of tools are available. If possible, we can make use of the tools through the mathematics of the that one I need to know, ma'am.
Rupali ma'am, please throw light on this, ma'am.
Actually, ma'am, maths is the basic of computer science. But
Yes, ma'am.
nowadays, students want to learn coding, but they don't want to do maths.
Yes, ma'am. Yes, ma'am.
Yes, they don't understand the importance of maths behind that coding, because
Yes,
maths is the basic, and without maths, their logic will not be get strengthened. Actually, I also feeling that, because we are running BCA course, BSc Computer Science course, but in the same things are here also. Students want to learn directly coding, but they don't have any interest in doing maths. They have a fear for maths. I don't know why it is that. We are also searching the solution for the same. How we
okay.
can create
Attract the
interest.
students.
Does create interest for maths within among student. So we are also searching, ma'am, for the solution.
Anyway, ma'am, seriously, I need to convey to the professor society that we are in a struggle now for all the mathematics people, seriously. So basic of all the subject, no, ma'am. So only it is reduced in all the areas, ma'am.
Yes, ma'am,
Seriously.
definitely, definitely.
Yes. You having any specialization, allied papers, mathematics, ma'am, in
Yes, ma'am.
computer science? You having allied paper mathematics in computer science, ma'am?
No, no, ma'am. No.
Mm. No paper in mathematics, ma'am.
No, no,
No.
no, ma'am. Actually, I am from computer science, and I have only a research in computer science only. No
Okay, ma'am.
paper from mathematics.
Anyway, ma'am. Okay, okay,
Okay.
okay, ma'am. In future, maybe there may be any collaborative
Yes,
we
ma'am.
can.
Definitely, definitely.
Okay, ma'am. Thank you. Thank you for it.
Searching.
Within a while, our speaker will be joining. Please check Rupali ma'am.
Yes. Sir is joining, ma'am.
Sir is joining. Yes.
Should I proceed?
No, sir hasn't joined yet, no? Please check whether has he joined. He will ping it as soon as he will join. Yogita ma'am, how many of the participants have submitted?
Ma'am, only 35.
Only 35. Okay.
Ma'am, may we leave or wait?
No, 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 ma'am, you please wait. Just a minute. Rupali ma'am, please see, sir said me that he is joining. Just
Okay,
called
okay.
me and said that he is joining.
No, ma'am.
Okay, then, ma'am. Okay.
1 minute.
1 minute.
No, there is no request.
1 minute, 1 minute, ma'am. Yes, send request, ma'am. Please just check, ma'am, please. Twice he has sent request, he is saying.
No, ma'am. No request.
No request. But he is saying that he has sent request.
एक और है. Yes, ma'am. Sir is there. Sir had joined.
Sir, please unmute yourself. Sir, please unmute yourself.
I'm sorry for being late.
Good evening, sir.
It's okay, sir.
Good evening, good evening. I'm sorry. Okay, yeah. Please, one second.
Yes. Good evening, everyone. I, Rupali Kharche, Head of the Computer Science Department and Co-Coordinator of this FDP, warmly welcome you all to Day 5 of the AICT ATAL Faculty Development Program on Technological Innovations in Agrotech and Food Processing. Today, we are privileged to have with us a highly experienced expert, Dr. Kishore Kumar Krishnani, who will guide us through two very important themes related to agri-market revolution and agripreneurship development. Our first session for today is on the topic climate smart innovations in agri-market revolution. For a detailed introduction of our distinguished expert, Dr. Kishore Kumar Krishnani, I request our coordinator, Dr. Shama Beg ma'am, to kindly present the profile. Please, ma'am.
Good evening, Rupali ma'am, and thank you so much. Good evening, sir. It is late, but you were busy somewhere and in meeting. And good evening to one and all present, the dignitaries, eminent speakers, participants, and distinguished guests to this AICTE ATAL Faculty Development Program. It's my privilege and I'm honored to introduce Professor Kishore Kumar Krishnani, my senior, my mentor, my friend, and a very good researcher also. The highest education is that which does not merely give us information, but makes our life in harmony with all existence, as said by Rabindranath Tagore. With this profound thought, it is my privilege to introduce our distinguished speaker, Professor Kishore Kumar Krishnani, holding a citation of about 6,168 presently, serving as Joint Director Academic, ICAR, Indian Institute of Agricultural Biotechnology, Ranchi, Jharkhand. Dr. Krishnani is an accomplished academician and scientist with over 31 years of extensive research experience, including his leadership as head of divisions at CIFE Mumbai and NIASM Baramati. He has taught a wide spectrum of subjects ranging from environmental biotechnology, nanobiotechnology, and molecular biology to omics-based approaches for agricultural sustainability, climate change, mitigation in farming systems, circular economy, phytochemistry, technology, commercialization, and IPR entrepreneurship. Being a scholar of chemistry, his teaching philosophy emphasizes integrative and applied learning, preparing students not only for academic pursuits, but also for entrepreneurial ventures and industrial collaborations. As a researcher, Dr. Krishnani has made pioneering contributions in climate change mitigation strategies for farming systems, circular economy approaches in agriculture, phytochemical applications, and integrated agri-aquaculture practices. His expertise in multi-omics technologies has opened new avenues in crops, livestock, and fisheries improvement, linking fundamental research with applied innovations. He has successfully built international collaborations with institutes in the USA, Australia, and the Philippines, contributing to cutting-edge agricultural biotechnology research and education. His significant work has benefited crops, livestock, fisheries, and integrated agri-aquaculture systems, especially for livelihood enhancement in tribal areas. Dr. Krishnani's academic contribution is vast, over 125 peer-reviewed papers, 20 book chapters, 350 gene sequences, and 12 patents, including 7 granted, along with the commercialization and relicensing of technologies. Professor Krishnani has guided numerous postgraduate and doctoral students, many of whom have gone on to establish themselves as successful scientists, academicians, and entrepreneurs. He has delivered more than 110 invited and keynote lectures across the globe, influencing both academic peers and young researchers with his vision of sustainable and innovation-driven agriculture. For his outstanding contributions, he has been honored with many prestigious recognitions, including the ICAR Rafi Ahmed Kidwai Award, NAAS NAS Biennial Recognition Award, Fellowships of NAS and BRSI, BHU Sentinel Award, Australian Government's Endeavour Fellowship, DBT Biotechnology Overseas Associateship at USA, Zika Fellowship at Philippines, and several Best Presentation Awards. In summary, Professor Krishnani is not just a scientist, but as a visionary academician, a thought leader whose work bridges research, innovation, entrepreneurship, and policy. His academic journey continues to inspire countless students, researchers, and professionals working toward technological transformation in agriculture and food systems. Indeed, Dr. Krishnani stands as a beacon of academic excellence, innovation, and global leadership in agricultural biotechnology. We are honored to welcome him today to share his wisdom and insights at his own place, Chhattisgarh, and he is an alumni of Government VYTPG Autonomous College, Durg, and also alumni of Chemistry Department of Chemistry, Pandit Ravi Shankar Shukla University. Welcome, sir, to your own place, Chhattisgarh.
Thank you, Dr. Shama. Am I audible?
Yes, sir, you're audible.
Yeah, thank you very much for the brief introduction. Not a brief, actually. At the outset, I'm sorry I joined late. Actually, I am in West Bengal now in Krishi Vigyan Kendra. There was some program here, and earlier I was in Meghalaya. Actually, I was supposed to deliver my talks, 2 talks on 18th of August. But that time I was in Shillong. So I'm sorry for that. So today I could find some time so that I'm able to deliver my presentation here. What I will do, I will present only one one particular topic. Second topic, maybe if possible, maybe tonight or maybe tomorrow early morning. Okay, I can make a presentation if possible, because this presentation itself will take maybe more than 2 hours. Okay, and when I was preparing this presentation, my file got corrupted. So again, I had to prepare. That is the reason it got delayed. Okay, so now, Dr. Shama, I have shared my presentation to you
Okay.
in your WhatsApp.
I'm sharing. Yes, sir. I'm
Please.
sharing it.
Hmm.
Okay, sir.
So quickly, total how many participants? I can see 62, right?
70,
Yes,
sir.
So I think that would, if I ask you to introduce, then I think it will take a lot of time. So maybe briefly, Dr. Shama, could you please tell me they are from which department and their faculty or student? That information, can you give it to me? Just briefly, you can give it to me. Dr. Hansa, how are you? Principal Madam, Namaskar. Madam, we joined here.
Yes, ma'am is here.
Thank you. Okay. My greetings, Madam. Namaskar. ठीक है, ओके। सबसे पहले तो बहुत बहुत धन्यवाद बुलाने के लिए मुझे, और मुझे अच्छा लगता है मेरी जो भी नॉलेज है शेयर करने में, तो थैंक यू वेरी मच मैडम, थैंक्स अ लॉट। ठीक है, Yeah.
Sir, many of the participants are from agricultural university, some are from colleges, and few research scholars are there, and few PG students are there, sir.
Okay, good. Okay.
And all over India, we have the participants all over India, from
Okay.
east to west and from north to south.
Okay, good. That's very nice. Okay. So please share my presentation, Dr. Shama.
Rupali ma'am, I have shared to you. Please start sharing.
Okay, ma'am.
Maybe some of the slides may not be in the proper order because I rearranged all the slides because my presentation got corrupted. So there was some issue like pictures were not showing. So good evening, Dr. Loknath.
Good evening, sir. Good evening.
Good evening. Good
Welcome,
evening.
sir.
Good evening, sir. I am Dr. Rupa from Ranchi University, sir.
Oh, Dr. Rupa Verma, how are you?
I am fine, sir. Good to
Great.
see you, sir.
Nice to meet you. That's very nice. Yeah,
Yes.
they invited me, Dr. Shama.
Yes,
Dr.
yes.
Rupa Verma, she invited me in Microbiology Day, International
Okay.
Day. I was
Okay.
a chief guest in Ranchi
Okay.
University.
Okay, okay.
So
We have
good.
added it in our report also, sir. And
Okay,
in,
Attached.
yes, sir.
that's very nice. Okay. Usually I don't give presentation unless I'm satisfied, but today presentation actually I have to re-prepare. So that is the reason I wanted to deliver my talk tomorrow early morning at 4 o'clock or maybe 3 o'clock. But it's okay. Whatever I have prepared, let's see. Okay. Yeah. So I will be presenting my second topic first. Agri entrepreneurship incubation in yeah. Please go to the first slide. Yeah. Please begin in the presentation mode. फुल स्क्रीन में लेके आ जाइए. नहीं, नीचे आपका होगा, वहां पे इसमें होगा कहीं ना कहीं फुल प्रेजेंटेशन बोर्ड में.
Sir, at your laptop, you have to move by yourself and the full
Is it?
screen
Is it?
to make it full screen.
No,
Yes,
I
sir.
do.
You just click it.
Okay. Is it okay now?
Yes,
Is it okay?
we are able to see it.
You are able to see. Is a full screen you are able to see or it's only half?
Yes,
Full, no?
yes. Yes, sir. From here, we are showing it in the full screen.
Okay, that's very nice. So, thank you very much once again for giving me an opportunity to deliver my talk on agri entrepreneurship, incubation, and agrotech and food processing. I'm going to share my experience of about 35 years, right from my joining at ICR, Indian Council of Agriculture Research, and also my overseas fellowship in 6 different state universities in the United States, Australia, and Philippines. So maybe it will be learning for me also. Maybe you can also share your experience so that I can also learn something from you. So, okay, next slide, please. Ours is Indian Institute of Agricultural Biotechnology, and it's a Ranchi hub that Rupa Verma, she knows very well from Ranchi University. And we have total 4 partners. IIB, Indian Institute of Agricultural Biotechnology, is the leader, is the main institute. And apart from that, we have 3 more institutes involved in the Ranchi hub. That is National Institute of Secondary Agriculture. Secondary agriculture means, like, you know very well that circular bioresource utilization, a lot of agro waste are generated. So how to utilize those agro waste material, okay? And even the lac research, there are many other things. They are doing a lot of research on that aspect. And apart from that, we have one more partner, that is NBPGR, National Bureau of Plant Genetic Resources. And also we have ICR Research Complex for Eastern region. The main institute is in Patna. They have the regional station here in the Ranchi. So they are our partners. So total 4 institutes are involved in our ICR IARI Mega University. IARI is an Indian Agricultural Research Institute, New Delhi, that you know very well, right? And we offer UG, PG, and PhD programs in different disciplines like molecular biology and biotechnology, genetics and plant breeding, biochemistry, and agricultural chemicals, and MTech degree in agriculture processing and food engineering. And also the doctoral program in the same disciplines we offer. And at present, we have about 120 students from all over country, about 19 to 20 states including Kerala, Tamil Nadu, and then Delhi, Haryana, Bihar, and West Bengal. Okay. Next slide, please. Okay. Before I go deep into entrepreneurship, let me tell you the 6th Dean Committee has recommended. Earlier we had a 5th Dean Committee. Now we have the 6th Dean Committee recommendation, where they have recommended that we should have the entrepreneurial development in biotechnology. So now I see even the national education policy, I think Dr. Sharma, she is the right person to tell us about national education policy. And because when I was discussing with her, even many things I learned from her when I was in group. Yeah. So even the national education policy says the same thing, like we should go for entrepreneurship development, multi-institutional and multidisciplinary work, we should do that, right? So that is the reason nowadays we are promoting the entrepreneurship development program, and even the 6th Dean Committee is recommending that. Next, please. Now, as far as entrepreneurship is concerned, so first we should know what is the actual definition of entrepreneur. So entrepreneur is an individual who accepts financial risk and undertake new financial ventures. Typically inspired to start a business because the entrepreneur perceives a consumer need, because one has to perceive a consumer need that is very much required, right? And also there are many other definitions have been given. This entrepreneur word, it has been derived from the French entre, and that means to enter, and the predent, that it means to take like to enter and to take, something like challenge or something like that, maybe financial risk. Okay. To help new technology come to the market, many universities have established business incubator. Now you can see we have total 113 ICR institutes all over the country. And in almost all the institutes, we have a research incubation center. Okay. And where what we do, we offer the services wherever you go, in Raipur, Ranchi, and because Raipur also we have an ICR institute. And if you want to become an entrepreneur in in agriculture science, so you can approach the nearby institute, ICR institute. If they have the incubation center, they will offer those services. Just you have to pay 5,000 rupees for 6 months. And then you can learn a lot. And even you can start entrepreneurship. I'm giving you these ideas because tomorrow if you want to become an entrepreneur, so definitely it will help you, right? So in ICR institutes nearby, you can go and find out if they have the incubation center. And there they will provide you all the services like equipment facility. They'll give you the training also. Like I have guided when I was in CIP Mumbai, I guided 3 entrepreneurs and for 6 months, and I have given them training on aquaculture and culture-based fisheries. Okay, so fisheries and integrated and aquaponics also, right? That's what I have done it. So that's the entrepreneur. Then next slide, please. Then entrepreneurship. The entrepreneurship, it was coined as this term in the second half of 18th century, long back. So entrepreneurship was there right from the beginning. Okay, so it is a process of action an entrepreneur undertakes to establish his enterprise. So many definitions have been given. You can see that it's a creative activity and it's an attitude of mind to seek opportunities, to take calculated risk. Okay, and a business plan or project, an initiative undertaken or something like that. Okay, and according to Peter, entrepreneurship is defined as a systematic innovation which consists in the purposeful and organized search for changes and the systematic analysis of opportunities such changes might offer for economic and social innovation. Now, you will be telling me, you will be asking me, Dr. Krishnani is giving me a very difficult definition of entrepreneurship, which has been given by Peter. Probably you would have seen that movie, 3 Idiot movie, right? In the 3 Idiot movie, like when the Amir Khan, he was asked to define the machine, then he has defined in a very simple language, and it should be like that. So you can see the entrepreneurship, it has been defined very nicely. But if you see the definition which has been given by Peter, that is very complicated, right? But you should know what entrepreneurship is. So that's the reason I have given you that movie example here, 3 Idiot movie example, okay? So next, please. Now, what is the process of entrepreneurship? Setting up of new entrepreneurship venture, and there are stages of Indian business or planning, right? Now, because now we talk about Make in India, we talk about Skill India, we talk about Startup India. These are the initiatives of Government of India, right? So there should be a self-discovery. Whatever you want to do, you should enjoy. If you really want to start something in entrepreneurship in agriculture, okay, you should enjoy. You should have some fire to do that. If you don't have fire, please don't do that. You'll miserably fail. So in the beginning itself, you have to decide. Now I'll give you my example. I have another 3 years and 6 months my service left. Probably I was planning to have start my some integrated farming in Chhattisgarh. So that kind of system, I'm very much keen to do that because it's very close to my heart. Okay, so here you have to enjoy doing it. Okay, and you have to examine the weaknesses. And if there are weaknesses, you have to convert the weakness into the strength. Here, I'll give you one more example. You have seen that movie Lagan, right? The Lagan movie, one actor, he was, when he was bowling, it was a spin ball, and he was a handicapped, right? So when he was a handicapped, when he was doing the spin ball, and that was his strength. The spin ball was his strength, but his weakness was his handicap. So he converted his weakness into the strength. That's what we have to do it. If definitely we may be having a lot of weakness before starting any entrepreneurship, but that weakness we have to convert into the strength. That is very much required. Then, yeah, please, the slide, please. Could you please go back to the yeah? I think it was the fourth or fifth slide. Yeah, this slide. Okay, fine. No. Previous slide, please. Previous slide. Previous slide. No, previous one. Previous one. Yeah, here. Then we have to identify the opportunity, looking for the needs, want, problem, challenges that we have to assess everything. Then we have to generate and evaluate the idea. That is very much required. You have to use your creativity and innovative way, right? If you really want to start some entrepreneurship, and then narrow the field to one best idea. You may be having a lot of idea, and out of so many ideas, you have to narrow down it, and you have to select any one particular idea where you are very close to that, and which you can enjoy doing it, right? Then planning, you have to do that definitely. Researching and identifying resources, and you have to raise the startup capital. Capital is very much required here. And that I'm going to tell you how to raise the capital in my next slide. And then launching the venture, developing a customer base, adjusting marketing and operational plan as required. And also the growth, okay, growth you have to see, definitely. Continuous monitoring is required, okay. And also one has to adopt the new circumstances. You have to see the ups and downs also. You have to expect that, and based on that only you have to adopt it. And then finally you have to harvest whatever, like say if you have developed some technology or something which you want to commercialize and bring it in the market, and which you want to promote it, you have to scale up the technology, right? And when you scale up the technology, the final product, you have to harvest it. So now you are ready to sell the business. And then for many entrepreneurs, you can see here, the new venture, new challenges will be there. So you should be ready for that. Next, please. And developing organization and management skill, here definitely the communication, delegation, vision, develop leaders, confidence, this decision making, that is very much required. I'll give you a few examples. I will not read everything here. Maybe a few examples I'll give it to you. Definitely you have to delegate power to your subordinate as well, because you see, you cannot be expert in everything. Say you want to have a microbial product, and that microbial product may be probiotics, which contains the bacteria. Now the bacteria, and also you want, it should also have some bioactive compounds, right? It should have the microbiome, it should have the metabolome, both together, and that should be immobilized on some matrix, right? So here you need a chemistry, you need a microbiology, you need a biotechnology. Okay, you cannot do everything. That is the reason when you hire someone, you trust him and give him the responsibility to do that. Let microbiologist isolate the bacteria, he will give it to you. Okay, you can immobilize it. Okay, and then you mix whichever way you want it, see the application. So that everything you have to see that. And also you have to motivate others, strategic planning, and you have to do the collaboration also, multidisciplinary work and multi-institutional, that is very much required. And also the initiative which you are taking it, and you should have trust on that, whatever you are doing it, definitely it will be successful. In the beginning itself, if you think that no, it is going to be fail, then better not to start it, right? So that is the reason, like if you see the developing organization and management skill, that is very much required. Next, please. Yeah, type of entrepreneurs here. Now you can see innovative entrepreneur. The best example, I will not define it. You yourself will understand that. Walt Disney, that's an innovative entrepreneur. Okay, even I have visited the Disney land in Orlando. I had been there along with my family. We had a very nice time. And it was way back, I remember it was 25 years back. See the advance, the technology, the science. Oh my God, I can't even imagine it. I visited as many as I think 2 or 3 parks in Disneyland, Disney World, and full of science, technologies were there. Okay. So it was there 25 years back. So that was a novel, innovative idea. And it's a huge success today, right? You know very well. And imitative entrepreneur, where you imitate others. Okay, that is also entrepreneur. They are also very successful. Okay, and the imitative entrepreneurs are also revolutionary and important because they are adaptive and they are more flexible. Okay, they'll imitate others. Where they are adaptive, they will say, okay, whatever technology other they have done it, I will imitate it. And that you can do it. In my next slide, I'm going to tell you, even someone has filed a patent in the United States, only in the United States, not in India. Then you can very well copy that work and you can commercialize the technology here. You can just Google search, literature search. Okay, any technology which is available, that you can very well take it. You can imitate the technology and you can develop it. And then Fabian entrepreneurs, they are shy and lazy, second generation entrepreneurs in a family business, just like I will give you one example like Mithai Wala. Okay, now that person, that maybe son or maybe daughter wants to continue the business, say sweet shop or maybe some other thing. Okay, so they are like Fabian entrepreneurs and they want to continue their family business and they're also very successful. Okay, they imitate only when they are sure that failure to do so would result in a loss of relative position in the enterprise. So that they take the calculated risk, right? That's why they do that. Then drone entrepreneurs who refuse to copy, they are recalcitrant, they are adamant. No, whatever I decided, that's my final decision. This is my technology, I will do that. Conventional way or whatever way. Okay. They are not ready to make changes in their existing production method, even if they suffer losses. They are ready to suffer the losses, but they will not accept any other suggestion. They refuse to copy. Right? They're kind of drone entrepreneurs. Next, please. Again, you can see there are different types of entrepreneurs here. Same thing only in agriculture science, in animal science, fishery science. Okay, business entrepreneur, veterinary. Okay, there are many others here. So same thing only. Next, please. Next, please. As I already told you. Okay. Then the entrepreneur definitely should have the vision, delegation, confidence, decision making, communication, risk taker, lead by other. He should be exemplary for others, and he should develop the leaders that should have good collaboration, adaptability, okay, and motivate others. Next, please. I think this I told you already. Next. Yes. Now I'll come to the agriculture science. Now you want to be an entrepreneur in agriculture science, right? Now, when we talk about agriculture, agriculture is not only crop plants, not only crops. It is animal, it is fish, okay? Altogether, the agriculture. When you do the fisheries, that is also agriculture. When you do animal husbandry, agriculture. When you do field crop or horticulture crop, that is also agriculture. Okay. So now here you can see the rice crop, that is a field crop. Okay. And apart from that, we have the horticulture crop, like say banana. Okay. Similarly, we have the poultry, we have the aquaculture fisheries, shrimp farming, crab farming. We have this goatry, duckery. Okay. So this one, in agriculture. Next, please. Now, as far as this agriculture products or agricultural entrepreneurships are concerned, There are different, the Indian biotechnology industry, you can broadly classify it into 5 categories here. Biopharma, like drugs, vaccine, diagnostics; biofuel, okay, nutraceuticals, enzymes; hybrid crop, biopesticide, biofertilizer, bioservices, and bioinformatics, right? And you can see there's a huge potential for this Indian biotechnology industries in coming years, definitely. An industry is expected to reach a market value of $300 billion by 2030. I came to know that probably it has already reached now. We are saying by 2030, maybe in 25 itself, it has already reached. That's what people are saying it. Okay. And it comprises more than 3,500 biotech startup and estimated to reach up to 10,000 by 24-25, maybe more than that, right? So that is the figure given by the DBT. So see the potential. Next. There are definitely a lot of career opportunities here in agricultural sciences, whatever entrepreneurship you do, whether it's related to your medicinal plant, bioactive compounds, okay, or maybe microbial products or nanobiotechnology, whatever which you apply in agriculture, whether in a crop, animal and fish, a lot of opportunities are there. Next, please. Here, the support for innovative technology development in India. Here, whatever like the product, suppose if you want to develop or if you want to have some entrepreneurship or a startup, there are many government agencies here, they will support you. You can see here, Department of Biotechnology, DST, and then also DSIR, CSIR, MSME, NRDC, DRDO, TIFIC is also there. And there are many others here. The international also, you can see there are some agencies whom you can approach, right? Next. Then SPERSH is under DBT. It's a social innovation program for products. Whatever product, if you are in your mind, which you want to commercialize or something, you have to give the proof of concept. Then funding support may be given up to 50 lakhs for a period up to 18 months, right? So it's the kind of startup you can very well do that. So financial supports are there, definitely. Okay. And the Indian academic scientist, researcher, PhD student, medical degree holders, biomedical engineering graduates, they are eligible to apply for that. Next, please. And funding opportunities, here again, the Department of Biotechnology, BIPP, that's the Biotechnology Industry Partnership Program. Okay. And that's again for R&D and the Small Business Innovation Research Initiative, there is IBRI, and Biotechnology Ignition Grant, also startup up to 50 lakhs, the DBT will give. Okay. You have to submit in the prescribed pro forma, okay, and based on that, if they find that it's innovative, and then they will definitely give you. Next, please. Then funding sources. Now, this is very important here because without capital you cannot do anything. Now, bootstrapping is like self. If you have sufficient money, you are a rich person, you can go for that. That is the bootstrapping. You can do that, right? Crowdfunding, investment from more than one person at the same time. Okay. There is crowdfunding. That is called crowdfunding. Angel investment, they are individual with surplus cash. If you have a lot of money, very rich person, and that money, like you want to simply spend it, okay, want to have some entrepreneurship or a startup, that you can very well do that. And venture capitalists, that's very important here. They are actually very rich people, industrialists, I should say, that professionally managed fund who invest in companies that have huge potential, right? So venture capitalists also, they can make investment. You can take a bank loan, even the business loan from microfinance provider, that is also possible. If you want to have this startup on a small scale, you can go for microfinance, and you can get money from friends and family and government grants. Next, please. Then component of business plan, you have to prepare a business plan. In the business plan, there's some document here. If you Google search, you'll get it. Okay, all this information you have to give. Okay, about the product and all those things. I'll give you an example here. Okay, in my next slide I'll give you. Okay, so here in the marketing plan, production plan, operation plan, manpower planning, organization, overhead charges, you have to include it here, including your own, the whatever time you're spending, your family, your children, they're spending time in an entrepreneurship, that also you should include it here in terms of money. That is called overhead charges. That is also equally very important here. Okay. So that everything you have to include and see, you have to calculate the cost benefit ratio, and finally you have to submit a plan. Okay, and maybe I'll give you some example where like how the cost benefit ratio is 4, even 5, even 10 also. Usually it may be 1.5 or 2. It can go up to 3, 4, 5, 10 also. You can see that. I'll give you a few examples here. Next, please. Now, develop leadership skill here, because that is very important here. Now, if you have decided that you want to be a agri entrepreneur and want to enter in agri entrepreneurship, so what you have to do it here, you should not be boss. You should be a leader. Okay. Leadership personality, you can see what Nelson Mandela says, it's better to lead from behind and to put others in front, especially when you celebrate victory. That's what happened with Dr. Abdul Kalam Azad, right? And you take the front line when there is a danger, then people will appreciate your leadership, okay? When any failure is there, you be in front. You take the responsibility. Yes, it is because of me. Because of me, it's failure. So that's what you should do that. If you don't do that, if you do the reverse, then people, they will not trust you and you will miserably fail. That is the reason you have to gain the support from your staff, your people, those who are involved in the entrepreneurship, right? So you have to develop a strategic plan from 3 to 5 years, communicate with transparency, share success, failures, spot and retain the best talent. That is very important here. I always believe in this. I always believe in this. Okay. I'll give you one example here, then you'll learn. I was participating in a research management position training program at NARM, Hyderabad, National Academy of Agriculture Sciences. And in the training program, they met some few groups. Okay. This is very important. Please listen carefully. They met few groups. In that each group, they asked us to, like, they have given us some points in terms of not rupees, some points. Okay. This much money you have, say you have 1 lakh rupees. Now that 1 lakh rupees you can spend for you developing the, like having the entrepreneurship. You want to start the business, you want to start up, we are giving you 1 lakh rupees, how you will spend it. So there were so many criteria, so many criteria. There was one criteria which I liked it very much, where we people become very कंजूस miser. उसमें हम सोचते हैं कि नहीं यार छोड़ यार क्यों इसमें इन्वेस्टमेंट करना छोड़ो और वहीं पे लोग गलती कर जाते हैं. Okay. That one particular point, there was one expert, he was expert in giving advice at particular point where this will fail. I'll give you one more example. I learned isolation of genomic DNA from bacteria, different methods. KIT method is there, CTEP method is there, chloroform method is there, okay? But I modified it, I developed my own. Okay. At one particular point, when you use ethanol, the ethanol traces will be there that will inhibit in DNA isolation. And that only expert can tell you. If you have to pay some money to expert to get that advice, pay him. And there, what we learned that many of us, we were like, why we should spend money on that expert, what advice he'll give. Okay. And later on, they miserably failed. They were not successful. Okay. Then that particular advice, when the expert has given, based on those who got the advice, they were very successful. So that is the reason wherever it is required, spend money. And not only that, I'll give you one more example here. I know many people, they miserably failed in their entrepreneurship. This once, twice, 3 times. And after 9th or 10th time, they were successful. And they spent almost 10 lakhs rupees, or maybe more than that. I was just discussing with them. You know, one of the guy, he was saying that whatever money I have spent 10 lakhs rupees for making this my entrepreneurship successful, that was a training program for me. I spent 10 lakhs rupees on the training program. I started my startup so many times, I miserably failed. So I was making investment in the training, I was learning. And that is, he was absolutely right. Okay. So like, don't get discouraged that if any failure is there, that is the training for you. Okay. And be exemplary for others. Seek advice. Good listener. One has to be a good listener. Okay. Develop leaders. Second lineup is very much required. Whatever you have acquired the knowledge, definitely we have to. Otherwise, if you don't write it somewhere, you see, how many years we will live? 100 years maximum, 125 years. एक दिन तो जाना ही है भाई, क्या करेंगे? तो ये सारे जो दिमाग में जो नॉलेज है, कहीं तो हमें उसको यूज करना है ना, किसी को तो देके जाना है ना, ठीक है ना? नेक्स्ट प्लीज. Yes, there are a lot of environmental factors here in entrepreneurship, especially in the agri entrepreneurship here. It all depends, but mostly that is related to the policy making. I'll give you some example. Coca-Cola company, I think I should not name it. Some company wanted to have some manufacturing unit in one particular state in South India, and like they wanted to start it, but they miserably failed because of a lot of resistance from the people. They were saying that it created a lot of pollution, so many things. And same thing happened with, very well, Nano car also, right? Tata's Nano car also, when he wanted to have it in West Bengal. So many things happened, right? So you see here, there are a lot of depends on the state government and also the policy making, political factors. Everything you should do the assessment first. Okay? You have to evaluate plus and minus. Then only you decide what next I should do that. Okay. Now I'll give you one more example. Sugar industry, right? When I was in Baramati, we did a lot of research on sugarcane, and a lot of sugar industries are there, factories are there in Maharashtra, and they are solely dependent on sugarcane cultivation. As a contract farming, they are doing it. Okay, you do the sugarcane, we will harvest and we'll take it in the sugar industry. It's going on. Okay, fine. It's a fully dependent on the sugarcane cultivation. Okay, but suppose farmers, they don't get the profit, then what will happen? You don't have any synthetic source where you can manufacture the sugar. That's not possible. You are purely dependent on the natural source only, that is sugarcane. Okay, so that everything you have to look into that and then decide. And also, I know, I'll give you one more example from Rananga itself. One professor in Rananga, he has started some fisheries entrepreneurship, and he purchased some, I think, 10-hectare land, but he miserably failed because the site selection itself was wrong. Water was contaminated. I came to know that there was a water logging always. So that was he miserably failed it. Okay, so that is the region you have to do the proper survey. That is very much required before making any investment. And then finally you decide what next you want to do that. Okay, and supply also you have to see. Okay, market trend, that is also you have to see. Next, please. Then definitely when you start your entrepreneurship, and you have to add the values within the problem. Okay, mainly you have to adopt it, and also you can see the adaptive and innovative problem solving that you have to do it. That is very much required here. Okay, and you can see the other, this I have been discussing with you. I think it's almost more or less same thing only, but you have to add the values in that, innovative or creative or adaptive. That is very much required. Okay, so be prepared for that. Maybe any problem you have to solve it. Next, please. Yes, now I'll start giving my examples here. Circular economy. We talk about the circular economy, right? Here, circular bioresource utilization. In, say, you have integrated farming system where you have the crop, you have the animal, you have the fish. Okay. Agro waste is generated from the crop plants, and that agro waste you can very well utilize for the compost. Okay. Organic manure or whatever. Right. And then when you are doing a fisheries, then water you can use it for irrigation in the crop plants, right? And you give a lot of feed in the fisheries. When you give a lot of feed has the protein has the nitrogen. So the water will get fertilized, and the fertilized water you can use for crop plants. Now you have the dairy there, buffalo or cow, whatever, the cow dung, the dung you can apply in the fisheries pond. So that will develop the primary productivity. Okay, that is required for the fish, very much required. So it's a kind of zero budget agriculture you are doing it. It's a kind of circular economy. It's a kind of circular bioresource utilization you are doing it, where the byproduct of one crop you are using for another crop. So nothing is going waste. And you are not buying anything from the market. Organic manure, now you have that. You're not buying fertilizers. Okay. Fertilizer water you're using for the crop, which has the nitrogen. You're using the cow dung for primary productivity development. Right? So in that way you are doing it. So it's a kind of integrated farming approach, which when you are doing it, then definitely it will be very successful. The circular bioeconomy is very much required for stress-resilient fisheries and aquaculture. It's not only fisheries and aquaculture, crop, animal, and fish. When we talk about this agriculture, crop, animal, and fish. Because usually what we do that in the circular economy, we have to reduce the waste, we have to reuse it, we have to recycle it, we have to regenerate it. There are 4 R years, right? There are 4 R. So that's very much required here. So this we have published a book chapter, so that it's available online. You can see that how the field crop, horticulture crop, kitchen gardening, and like animal husbandry and fisheries with the we are done. We are given that. Next, please. Now I am coming to $1 million industry. I should say that I was talking about cost benefit ratio. No, 2, 3, 4. Okay, it's possible here. I'll give you the simplest example here. Now, we have successfully demonstrated integrated agri-aquaculture in Nandurbar area in Maharashtra. They were doing only field crop, mostly rice. We integrated the horticulture crop. We integrated the animal husbandry, dairy and goatry and poultry. And also we integrated fisheries was very important here. The main problem was the availability of the fish seeds, from where to get it. So we approach in the border area in Gujarat, Okai Dam in Gujarat. We purchase seeds, you know how much? एक मछली का बीज हमें कितने का मिला? 25 पैसे का, only 25 पैसे, maximum 50 पैसे. एक मछली का बीज 25 पैसे और 50 पैसे. When you culture this, when you stock in the pond, after 1 year or so, or maybe after 10 months, when you are giving feed and everything, okay, that one seed will become 1 kg, right? Here you are giving feed, protein feed, right? Fine. And you are doing water and whatever, okay? Overhead charges, many other things. Let us calculate maybe around 25 rupees you are spending to produce 1 kg fish. Today, Rohu Katla Mrigal, Indian major crops, IMC, in the market when you are selling it, minimum 150 rupees. Because I live in Ranchi, so I go to the fish market in Ranchi there. I am buying that fish not less than 200 rupees. Big fish, if you want to buy it, it goes up to 270 or 300 rupees. The same Rohu, I tell you. Okay. So minimum 150 rupees it will be there. And here how much you spend? 1 kg I'm talking about. Only 25 rupees. Let us take on higher and higher and higher side. There was some 10% mortality was there. 50 rupees maximum. Still you have 150 rupees. Right? So it is 1:3 3 cost benefit ratio. 50 rupees you spend, you are getting 150 rupees. So here, the freshwater aquaculture. Okay. Then we have the brackish water aquaculture. We have the inland saline aquaculture. We have the culture-based fisheries, reservoir fisheries. Okay. But since we live in the era of climate change, definitely what we have to do, we have to go for climate resilient agriculture. If you want to have any entrepreneurship, you have to go for climate resilient agriculture. That is required. You know why? You see, temperature is increasing, chemical pollution. My next presentation, maybe tomorrow or today, is not possible. I'm going to tell you, I'm not going to frighten you, but I'm going to give you a real figure what's happening in America. Okay? How much chemical pollution. So please don't miss that my presentation tomorrow. Here, the chemical pollution, temperature, salinity is increasing, salt is increasing. Okay? You know, in India, at present, we have about 10 million hectare area which are affected with the salt. 10 million hectare area, which we are expecting by 2050, it's going to become maybe 20 million hectare area, double. जब हर जगह साल्ट हो जाएगा तो एग्रीकल्चर कैसे करोगे? करेंगे क्या? ठीक है ना? तो क्या करें इसके लिए? If you go to Haryana and Punjab, already most of the places have become salt affected area. You know what they are doing it now? They are doing brackish water aquaculture there. That is called inland saline aquaculture. Because in Haryana, Punjab, you don't have any coastal area. It's an inland area. Fresh water was there, got salinated now. The saline water, now you are culturing the fishes, which you culture in the coastal water. So now you're doing an inland saline aquaculture here. Right? Now, like fishes, I'll give you example. Milkfish, chanos, mullet, mugilcephalus, Lattice gallicaribus, sea bass. These are the coastal aquaculture fishes. In sea water, you have to culture. But now they are culturing it in inland saline water in Haryana, Punjab. Okay? And these are the fishes which are euryhaline fishes. You know, euryhaline fishes means, one second, I have to bring my, hold on. My laptop, I had to put the charger here. Okay, maybe 1 or 2 slides. Maybe we can have some break for 2 or 3 minutes. I'll bring my charger, laptop charger. It is in my room here in guest house. Okay, so that's inland saline aquaculture, right? And then we have the culture-based fisheries. Culture-based fisheries means, now you see, China is using every single drop of water. If there is a rain, there's some water logging area, they will simply stock the fish, say 8 months, 9 months, and they'll harvest it. Wherever small reservoir is there, simply we can stock the fish. You don't need to give any feed. After 8 months, 9 months, 1 month, simply harvest it, sell it. That is the culture-based fisheries, reservoir fisheries, you can do that. There's no investment is required. Only you have to buy the seeds and you do the stock enhancement. Just fish seeds you have added. Okay. Now we do in the Ganga River, no stock enhancement, we are doing it, right? You might have heard about it. To like the fish, we have to put it in the Ganga River and other things, right? So other rivers also. So that's what we have been doing it. So here, the culture-based fisheries is equally very important, but simultaneously, what you have to do that you have to go for the climate resilient species. It should be air breathing, salinity tolerant, temperature tolerant. Tilapia fish, you know very well, it's a tolerant. Maharashtra people, they eat a lot of tilapia. And then also the shingi fish, there are many other fishes which are air breathing fishes. Okay, so that can be stopped. Right. So I think we can break for 2 or 3 minutes. I'll bring my this one.
Sure,
Chart.
sir.
Okay.
Sure,
Okay.
sir. Sure.
Yeah.
Participants can type the questions till that time. If you have any queries,
Yes, I'm back. So, should I continue, or if you have any query, you can ask me?
Yes, sir, you can continue. And there are, yes, sir. One question is there, sir, that so at the very beginning you mentioned agro waste management. So kindly share your experiences on parali, remaining part of rice crop after harvesting, management using biotechnological and microbiological methods. Is
Okay.
there any pre-existing method available for the same?
Okay.
Dr. Loknath Deshmukh is asking,
Yes.
sir.
Dr. Loknath, I appreciate you for asking this question that was very much required. Okay. So, actually, parali, like in Haryana, Punjab, another state, okay, they will simply burn it. Okay. And it is creating a lot of pollution, especially in the Delhi. So, parali, actually, what they do that, parali can be used as a mulching. You can use it as a mulch. Okay. What usually we do that for mulching, we use the plastic, right? And plastic, again, that we are facing a big problem of microplastic nowadays, right? That I'm going to tell you in my next presentation tomorrow about microplastic. But here, this plastic, we can avoid it and we can use it as a mulch. That one thing. And second thing is the work which I have done it, that is on why this rice straw or the parali, whatever, there, the paddy straw, there here, why there's a recalcitrance of this material. The recalcitrance is because of lignin. Okay. And the lignin is the material which is chemical which gives a lot of recalcitrance to the plant, right? And the lignin has to be degraded. So, like many researchers, what they have done it, including me, we had done some work on isolation of lignin degrading bacteria. When I was some CSR of Canberra, I did some work on that. Lignin degrading bacteria. Okay, in Baramati also we tried some work, we did it. So if you are able to degrade the lignin, then definitely it can be degraded. So it is a biodegradable material, but recalcitrance is there because of lignin. If you are able to degrade the lignin, then it can be degraded. So that may be possible. So either it can be used as a mulch in the next crop, or it can be degraded using microbes. Yeah. Yes, Dr. Loknath, is it okay? Or but at present, you see, many researchers they have claimed that they have developed some microbial formulation, microbial product, which can degrade it. Okay, the parali. And but you see, like when you go for burning, it's an easy way of doing it to eradicate it, get rid of that. So here, but lignin degradation, if you want to do that, it will take some time, right? So that's what biodegradation or maybe mulching.
Thank you so much, sir. Thank you so much, sir. It's wonderful, sir. Thank you.
Okay. Yes. Any other question? Okay. Then I'll move to the next slide. Next, please. Now here, the species diversification, whether it's you call it a crop diversification or species diversification. If it is a plant, then definitely you have to go for plant diversification, crop diversification. Okay. Now I'll give you this example of fish here. Freshwater aquaculture we do. We have the freshwater fish. We have the freshwater prawn. Okay. Like we have the IMC, Rohu Katla Mrigal, and we have that Rosenbergi, Macrobricum Rosenbergi, that is the prawn. Okay. And then we have the other freshwater fishes here. Here, definitely the species diversification, the need of our today, because you live in the era of climate change induced abiotic and biotic stresses, especially in the inland side aquaculture. Right? Other side, you have the brackish water coastal aquaculture fish and shrimp. Okay, the species diversification, I'll give you one example here. Here, even one fish which is cultured in coastal water in a high saline area. When I say coastal water, can anyone tell me in 1 liter of coastal water about how much salt is there? If you take 1 liter water and if you evaporate it, how much quantity salt you'll get? You can tell me in the range also, that will do. Anyone? You can give me a range also. how much quantity salt you will get if you evaporate 1 liter of water. Okay. 1 liter coastal water, if you evaporate, you may get 30 to 35 gram of salt. Okay. Let us say 35 gram. So that 35 gram salt, fish which you are doing a culture, now all of a sudden, if you bring it at the 0 ppt, no salt, that is also surviving. That's what we have done the work when we were in the Mumbai. It depends the fish how it adopt it. Even fish can adopt high saline area to low saline area. But that you have to do in the gradient manner, slowly. If 35 directly to 0, fish will die. 35, bring it to 30, 25. That is innovation. That is the novelty. Suppose tomorrow, if you are hatchery, if you have the fish pond, and if you want to culture the brackish water fishes, coastal water fishes, the fish seed, you'll get it from Tamil Nadu or some other part of the country, coastal, where the brackish water aquaculture is going on. Those fishes, you are getting it here. Okay, and you want to culture in 0 ppt because, say, in your state, you don't have the coastal water. It's not coastal area. You have only fresh water, but you want to have that fish because that fish is high value fish. Most of the marine fishes are high value. It goes up to 1,000 rupees per kg. Am I right or not? So that fish you want to culture, say, in your area, where, like say in Chhattisgarh, no coastal area. Jharkhand, no Chhattisgarh, no coastal area. There you want to do that. You can very well do that, but slowly, you have to reduce the salinity gradient and adopt. Once it will be adopted, then even 0 ppt, 0 saline water, you can culture it. And it's growing very well, I tell you. That's our experience. And this is called species diversification. This is called your innovation. And let me tell you, if you are able to do that, and if you are successful, today, Hilsa fish, how much it is sold in the market, 2,000 rupees per kg. But the problem with Hilsa fish is, it's a migratory fish. It will migrate from fresh water to brackish water for the breeding, for the seed production, seed generation, for the breeding purpose. It will move from fresh water to brackish water. That's what's happening. Okay. So, but it's a high value fish. Next, please. Here, now we talk about World Health, whether it's a United Nation or FAO, we talk about the One Health, right? And this is the only, I think it's the best solution for today's problem, because the way the climate change is there, many stresses are increasing, abiotic stress and biotic stresses, and One Health may be the solution. I will give you one example here. I had been to Digvijay College in Chhattisgarh because I belong to Durg, Chhattisgarh, okay, Durg Bhilai. I was born and brought up in Durg Bhilai. So often they used to invite me in Bhilai, Durg, Chhattisgarh, so I used to go there. So then in Digvijay College, one faculty, he gave me one medicinal plant. And he told me, sir, this I have collected from jungle. और जंगल भी बहुत अंदर जाके जंगलों में जाके लेके आया हूँ. बोला सर इसको आप पहनिए गले में. मैं पहनता हूँ कभी कभी, अभी घर में रख के आया, अभी तो बाहर आया हूँ. तो it controls the BP. Unbelievable, really.
Yes.
Yeah, that's the medicinal plant. That's the beauty of that. That's the bioprospecting you are doing it. That is called ethnobotany. So go to the jungle, discuss with tribals. They will tell you, they don't know the chemistry of that medicinal plant, but they will tell you this plant they are using for that particular disease, for remedy. Okay. Now take that plant, do the chemistry, do the bioprospecting, do the bioactive compounds. Okay. And then that's what, like, you can do wonders, I tell you, because you live in the era of antimicrobial resistance today. What are antibiotics we eat not working? Okay. Then definitely we have to look for some new antibiotics, plant-based antibiotics from medicinal plant. Today we talk about the neem. Why we say neem? We are using it because neem produces more than 500 bioactive compounds. That is the reason neem plant is the potent insecticidal. It has got the 500 insecticide molecules. Okay. Suppose one molecule is developing a resistance against any insect, pest. Another 499 molecules are there. It will take more than 200 years. Okay. So that's what I'm saying to you. Here, bioprospecting, that's a part of One Health. Because when we say One Health, definitely you have to take care of human, you have to take care of environment. Environment is very important here, and you have to take care of agriculture. Agriculture, when I say, it's a plant and animal both. Okay, so you have to take care of everything together. Okay, so here the bioprospecting and microbiome and metabolo. Again, I tell you, I had been to Philippines on some Japanese fellowship. So in the mid of the sea, we were taken there, and then we did the snorkeling there. And I was able to see a lot of marine organisms, inside. I was able to see that. And if someone goes inside deep in the sea, collects some coral reef or some marine organisms, I'm pretty sure you will get a novel microbiome and metabolome. Try that. I'm giving a wonderful research idea. Okay. So that bio, and then once you are able to get some bactericidal chemical or maybe some antibiotics, novel antibiotics, once you got it, determine the structure by NMR and other chemical, other methods, instrumentation, sophisticated equipment like NMR and other things, LCMS, GCMS, whatever you want, you can do that. Okay, XRD, FTIL. And then after doing it, once you know the structure of the molecule, you can synthesize. That's what antibiotics we are getting it in the market. They are synthetic. How they got the antibiotics in the market, synthetic? Because from the natural source only. From nature they got it, they isolated it, they determined the structure of the molecule, and then they synthesize in the lab. That's why they are getting antibiotics here in the medical shop. Okay. And then microbiome is a good source of bacteria. Okay. And those beneficial bacteria. I think Dr. Shima will be able to tell you about 95% bacteria are still unculturable. Only 5% are culturable, right?
Yes.
Earlier we used to say that 1% are culturable, 99 are. Now we say that 95% are unculturable. And that is very much true, yes. So 95% you see the microbiome hidden. That you have to tap it. You have to tap it. Okay, the huge potential is there, huge. Okay, that's
So
what
one thing also I want to say that in between that about medicinal plant, what you are saying that we should get their what chemicals are present there or what molecules are present in that.
I'm working on that.
Our novel research says that those molecules or the secondary metabolites are because of the presence of endogenous microorganisms which are present inside the plant, and they are not harming the plants but are giving the medicinal properties to that plant. So.
Exactly, you are absolutely right, Dr. Shama, because here there are rhizospheric bacteria in the plant root zone. You'll find the bacteria which they are supplying nutrients to plant.
Yes.
Okay, the beneficial bacteria. So bacteria are supplying nutrients to the plant. Bioactive compounds they are supplying to the plants, right? So
Yes.
that's what there's an association, symbiosis between bacteria and the plant.
And the plant.
You are
yes.
absolutely right. Yes, you are correct.
Yes.
So here, those microbiome, whether in the rhizosphere area or whatever, okay, that we have to definitely tap it. That is very much required. Okay. And then plant growth promoting bacteria. There is a huge market for plant growth promoting bacteria, I tell you, huge market. If you are able to isolate any bacteria which is plant growth promoting, simply immobilize, develop a product, develop a microbial product and sell it in the market. But it should be novel and innovative idea. Okay, those who are others who are not given it. That's what we should do that, right? So here again, same thing only. Okay, integrated. Next, please. I already told you about the circular economy, integrated farming, and here the integrated agri aquaculture. Okay, because you can see the use of animal manure as a pond fertilizer, the use of crop byproduct as a supplementary feed for fish, the use of pond sediment as a terrestrial crop fertilizer, the use of aquaculture wastewater for crop irrigation. Okay, there are so many things here, and you can very well do that, right? Here, the linear economy was take the raw material, develop a product, distribute it, consume it, and simply dispose it out. That was a linear economy earlier. Okay, lot of waste material going to the environment, causing lot of environmental pollution, linear economy. But now we have to do the circular economy. Nothing should go waste. Sugar industry is the industry which is a zero waste industry. Everything is fully utilized. Bagasse, you are using it. Molasses, you are using for winery. You are using everything. And there is a huge demand for the byproduct of sugar industry, I tell you. Okay, next please. Yes, the nutritional biotechnology also is equally important now. The way your protein sources are getting depleted, what will you do? Definitely, we have to go for microalgae, right? Microalgae is the solution. We have to go for the probiotics. We have to go for the adjuvants. We have to go for nutraceuticals, feed ingredients, additives, immunostimulant, biofortification. That's what we should do. That it's a microalgae. Or maybe I tell you, I was in Jabalpur in Directorate of Wheat Science Research, Jabalpur Wheat Institute, and there I used to say that why wheat means what? Unwanted plant. We don't need that. I used to tell them, I think we should change the name of the institute. It should not be Wheat Institute. It should be plant. All the plants are valuable. Even though wheat, even the wheat plant, you can use it somewhere. It's a good source of your bioactive compound, your metabolome, right? It may contain a lot of bioactive compounds. Okay, so that is the region now today when the wheat are the valuable source. Iconia, water hyacinth. Okay, if you go to the Maharashtra side near Dawn area, between Dawn and Pune, you will find one water body, not via Pune, Dawn, if you go towards this one, some other place, there is some small water body there. There Iconia, I could notice that. Not Iconia was there, water hyacinth. So that water hyacinth, you can use it. Even we use the water hyacinth for fish breeding, you know that? For fish breeding, some plant material is required as a substrate for the fish. Okay, so that's what. So see the application, right? That's what we should do that. And then prebiotics also definitely is a huge market for that, because definitely you need the prebiotics for developing the probiotics. That is very much required, right? Okay, then next page. As I told you, my few slides got corrupted, but so I had no time. So that is the reason I think you're not able to read it. But anyway, I'll try that. Aquaponics system. Now, aquaponics, when you do, here you do the fish culture along with the plant. We have published many papers in 2025 related to aquaponics. You can see about, I think in 25, we have published about 20 papers in a high-paid journal. Aquaponics, biofloc. There are so many things we have done it here. And also like in the biofloc system, you can see. So here, the aquaponics, when I say, you are cultivating plant and fish, right? Fish water you are using for plant, recirculatory mode, continuously recirculating it. Nothing is going waste. Okay, that's what you are doing it here. So here in the aquaponics, in the plant, you are using the soil. But in the hydroponics, you don't use the soil. Direct in the water, you do the plant. Okay, and fish. But here in the aquaponics, you are having the plant in the soil and the water. I mean, like in the fish. So that's what like aquaponics. Even you can do in the roof terrace there, the top in your apartment. That also possible. Okay, and it's gaining a lot of momentum nowadays. Aquaponics is a successful technology, I tell you. Even I know Dr. Shama, she knows very well. I have one of my friend, Dr. Harnam Singh Alreja. He lives in Bilai, and he's doing all of the terraces. You can see that all this not fish. He's a vegetarian. He's not doing fish culture. He's not doing an aquaponics, but he's doing all of the plant, vegetables, everything. He's doing it. Organic farming. See that in the terrace, in the apartment, he's doing it. In his own house, he's doing it. Okay. So then again, pearl culture. Pearl culture, Hyderabad is the hub, you know very well. Pearl is produced by the mussel, you know, one organism, freshwater organism. Okay. And then you have to develop a nucleus, put it in that animal. Okay. And then it will take time. So I know one entrepreneur, his name is Ashok Manwani, and he lives in Varanasi. Just Google search. And he lives in Banaras Hindu University, where he has got some space where he is doing all this pulp farming. And the pulp farming, he is a very successful entrepreneur. And he got the training from ICR Institute for pulp farming. And the pulp farming, recently we invited him as a professor of practice, where we were conducting one brainstorming session on emerging contaminants. We invited him. So online he participated. And you know what he was telling? Earlier he used to get pearl within 1 year. Now it is taking more than 2 years to get one pearl. It's all because of environmental pollution, which I'm going to tell you in my next presentation tomorrow. Okay. So here, This pulp farming is equally very important here. Okay. And if you have the expertise, definitely you can do that. An integrated multi-trophic aquaculture, where you can do all this fish shrimp farming together. Algae also you can do that. You can do the aquatic weed, aquatic plant you can do together. Aquatic plant, fish and shrimp or prawn together you can do that. Okay. So it's a kind of integrated multi-trophic aquaculture can be done. Next, please. And this I already told you. Next. Then yes, there's a huge potential. This probably I think I should tell you tomorrow, but I should give you some brief idea about that. This is also a big industry today. You know, in United States, where I had been to US twice, and then Australia also once. So there, you know what they do, that if any chemical pollution is there in the water, and that mostly coming from the agriculture, pesticide or whatever, it is contaminating the groundwater, leaching in the groundwater, and the groundwater, what they'll do, they will pump the hydrogen in the groundwater, and then there are anaerobic bacteria in the groundwater, and they will enhance the growth of those facultative anaerobic bacteria through maybe oxygen or whatever. Okay. Because, sorry, if they're anaerobic, you don't need oxygen. You need only hydrogen. They supply the hydrogen or something, some other gases, not oxygen. And anaerobically, they will enhance the growth of the bacteria. And bacteria, once the growth increase, they will degrade all the chemical pollutant in the groundwater, all pesticide. They are gone up to that much level. It's $1 million industry today, and that is called environmental biotechnology. So entrepreneurship can also be possible here in environmental biotechnology. Next, please. Then these are the bacteria, real images which we image by scanning electron microscope. You can see the lot of bacteria in the first slide on top one. The right side you can see single Pseudomonas aeruginosa, and left side you can see two Bacillus subtilis bacteria. See the size less than 1 micron. Okay, and which we image by scanning electron microscope. Microbes, definitely, I tell you, they are not only always pathogenic, they are beneficial as well. So those beneficial bacteria can very well be used. You can develop a microbial product, immobilize the bacteria, okay, and keep it for 1 year and then you can sell it. That's what you can do there as a probiotics or whatever. Next, please. Then again, as Dr. Loknath was asking about the agro waste material, there are a lot of agro waste material generated not only from rice. We have the crop like food and fodder. We have the plantation bioenergy crop. We have the medicinal aromatic plant. We have the livestock, dairy, poultry, okay, piggery, cash and oilseed crop, horticulture crop, aquatic and terrestrial wheat, poultry, duckery, and fisheries. Okay, a lot of waste material are generated from this, and those waste material can very well be used for bioremediation, for removal of heavy metals. Okay, and so that is also a possibility. Next, please. As I told you, we live in the antimicrobial resistance era now. Okay, whatever antibiotics you are eating, taking, not working. So definitely we have to do something. Even someone has told that we should make peace with the bacteria. भैया हम हार गए, आप जीते भैया, बैक्टीरिया आप ही राजा हो, आपके साथ मिलके रहना है, हमें अब ये हालत आ गई है. ठीक है, ये मतलब होने जा रहे हैं, मैं बता रहा हूँ. तो बैक्टीरिया के साथ खुशी खुशी रहिए. ऑलरेडी देख लिया आपने, कोरोना पेंडेमिक का टाइम देख लिया आपने. ठीक है ना, तो अभी तो पता नहीं क्या आएगा, भगवान जाने. लेकिन ये है कि ये मतलब चलते रहेगा ये, उसके लिए हमें तैयारी करनी पड़ेगी. हर चीज के लिए कुछ ना कुछ करना पड़ेगा. बायो प्रोस्पेक्टिंग है, मेडिसिनल प्लांट है, ओके, बहुत सारी अपॉर्चुनिटीज हैं. देखिए, एक चीज मैं कहना चाहता हूँ, जहाँ पे प्रॉब्लम्स है वहाँ पे सॉल्यूशन भी है. मैं हमेशा ये सोचता हूँ कि प्रॉब्लम बाद में आता है, सॉल्यूशन पहले से ही तैयार रहता है. मैं इस मामले में बहुत पॉजिटिव थिंकिंग रहती है रिसर्च के मामले में. सॉल्यूशन है, पहले से सॉल्यूशन है, प्रॉब्लम बाद में आएगा. तो हर चीज का, हर प्रॉब्लम का कहीं ना कहीं एक सॉल्यूशन जरूर होता ही है. ठीक है ना. नेक्स्ट प्लीज. देन आप इसमें देखेंगे कि एक फिश लाइन, मैं अभी इसमें आता है, बहुत इंपॉर्टेंट चीज मैं आपसे शेयर करने जा रहा हूँ. यू नो वेरी वेल, फिश प्रोड्यूस द स्लाइम, ग्रीन स्लाइम, है ना, म्यूकस होता है ना, फिश को पकड़ने के बाद स्लिप हो जाती है. फिश म्यूकस, ग्रीन स्लाइम, ओके, दैट इज the self-defense mechanism of the fish. When fish is under stress, it will produce more green slime. Mucus is a self-defense mechanism. हर ऑर्गेनिज्म का एक सेल्फ डिफेंस मैकेनिज्म, हमारा भी होता है, हमारा भी पसीना आता है, बहुत सारी चीजें होती है, तो ये सारी चीजें जो है सेल्फ डिफेंस मैकेनिज्म में आता है। तो हमने काफी काम किया था उसमें। तो नेक्स्ट स्लाइड प्लीज, मैं आपको जरा क्लियरली बता पाऊंगा नेक्स्ट स्लाइड में। नेक्स्ट स्लाइड, नेक्स्ट करिए, शायद मिस तो नहीं हो गया। नेक्स्ट 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 करते जाइए, मैं आपको स्टॉप कर दूंगा। हाँ ये यस 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 प्रियसन प्रियसन, नहीं नहीं आगे आपने स्लाइड दिखाई देना, हमें इसके नीचे जाइए। हाँ ये वाला, हियर यू कैन सी हियर, आई विल गिव यू एन एग्जांपल हियर व्हाट वी डिड। This technology actually I learned from Philippines. What they used to do, they integrate shrimp farming with the fish farming. Okay. Now the shrimp farming, you know very well, is $1 million industry. Mostly we are doing export. Okay. झींगा जो आप खाते हैं shrimp farming, lot of disease आती हैं, Vibrio harvey, एंगुलरम एरोजनोसा, बहुत सारे दूसरी भी स्यूडोमोनास एरोजनोसा के बहुत सारे बैक्टीरिया की डिजीज आती है उसमें। मैं ये कहना चाहता हूँ कि what we did it here in the center of the shrimp pond, we stock the one pen, mosquito net, that is called pen, and inside the pen we put some herbivorous fishes. Herbivorous fishes which are not carnivorous, which are, you can otherwise you can say that vegetarian fishes. So herbivorous fishes हमने stock किया. Those fishes, they produce the green slime, and the green slime is the excellent source of microbiome and metabolome. Produce lot of bioactive compounds because it has a lot of microorganisms, beneficial bacteria, and those beneficial bacteria, they are antagonistic to shrimp pathogenic bacteria. So in that way, it's a kind of green water technology which we have successfully developed in a coastal area. What we did further work on that. We apply the metagenomics and we examine the bacterial diversity of the green slime. Not only that, we isolated those bacteria and also we isolated the bioactive compounds from those bacteria also. So that paper will be published very soon in Microbial Pathogenesis. So here is a good source of microbiome and metabolome. So here those molecules which are present in the green slime, natural antibiotics, which are the natural probiotic bacteria present in the slime. No one has worked it. Okay. We have about 3,000 fish species in India. है कि नहीं? कोई भी fish ले लीजिए, ठीक है? तो यहाँ पे देखिए इनके molecules, fish लिया, then bacteria, then biomolecules, then zone of inhibition, it's antagonistic, right? Then shrimp farming. So in that way, we have successfully demonstrated the green water technology. It's all because fish mucus. There's a good source of microbiome and metabolome. ठीक है? Next page. बैक करिए. Next. Yeah. Now I'll come to nanotechnology. Now you see, even our body produces nanoparticles, I tell you. There is also self-defense mechanism in our body. And nanoparticles are bactericidal, good for bactericidal agent, especially the silver nanoparticle, right? So nanotechnology, nanoparticle, here, a lot of potential here to apply in agriculture. If you are able to develop any product, nano-based product, which kills, say, pathogenic fungi, pathogenic bacteria, pathogenic viruses, it's antimicrobial, for example, that you can very well use it. And not only in crop plants, but also in animal, but also in fish, right? That's a nanotechnology. So here, you can see here, the real picture are there, nanoparticles you can see. Left-hand side, you can see the small tube, that is the carbon nanotube, that is the carbon nanotube which was imaged by atomic force microscope, real image. And then two more pictures you can see here, that is the nanofiber. Okay. Next, please. Then again, it's a carbon nanotube imaged by atomic force microscope. Next. Yeah. Here, now say pandemic time, corona. Now you want to have some method which can quickly tell you whether it's a positive or negative. Okay, I'll share one example with you here. During pandemic time, we got, I think, almost 2 months leave because we were asked to sit at home, work from home. And my younger son also, he was staying with me in Mumbai in that on-campus accommodation, the stop quarter there. And my son, he was reported positive. Okay, he was diagnosed positive, and but I was negative. Okay, I was living with him. But he was very happy, he was smiling, and like just some fever, he got it for 1 or 2 days. So well, I'm a researcher, even I was doing research on my son. Then we gave the sample and the real-time PCR, we did it. Okay, the CT value was, CT value was about, say 23 or 24. Okay, I was quite confident that nothing is going to happen this fellow because CT value is 23, 24. If it was 5 or 4, like it would have been critical. Okay, but it was 23, 24. I was quite confident nothing will happen to him. Within 1 week or 2 weeks, again we did real-time PCR. So now CT values have gone up to 30, but positive. So it means he's recovering. Then in the third test, he was negative. Maybe it would have gone 35 or 36. 36 CT value means it's reported as a negative value, whatever. He recovered. So here, but real-time PCR, he was coming, taking sample and giving after results after 2-3 days. Here, if any sensor technology was there, I would have immediately used a sensor, some chip, some device. You can see here in the slide, chips. Immediately, I would have come to know that whether it is positive or negative within few minutes. Whether is it possible? Yes, that's an innovative idea. We can do that. Okay, lot of sensor technology. We have published one paper in a high-paid journal. There's a review paper. You can go through it. You get a lot of idea. Pooja is my PhD student. She got degree awarded last year. So this technology are cost-effective, rapid, okay, biocompatible, portable, where you use analyte. Analyte means say maybe anything, maybe virus, bacteria, or chemical pollutant, anything, biological element, you have to use it, okay, maybe antibodies, okay. Suppose if you have the virus, it's an antigen is the virus, antibodies, you have to raise it, so it's a biological element. So any signal, when there is a reaction between antigen and antibodies, always there will be some signal will be there. Maybe that maybe in terms of maybe heat, increase in heat, pH change, or maybe that electricity, current, okay. So based on that, you can very well detect, you can quantify how much viral particle or bacterial particles are present in that sample, okay. So that's a sensor technology. It's a huge scope for developing the biosensor, okay. But you should know the nanotechnology here. Not only that, here your chemistry and microbiology is not going to work. It will work. It will work up to 75%. Here you need electronics, physics, minimum 25%. Without electronics, physics, you cannot do anything. Okay. So that's what. It's a multidisciplinary work. Next, please. Many papers we have published online. It's available. You can see related to nanotechnology. Next, please. Application also. Okay. And then you can see here. I'll just, okay. Next. Coming twice, I think. Okay. Silver nanoparticles and all those things. Next. Okay, next. Okay, हो गया already कर दिया. Next. बताइए. हो गया, सब हो गया. ये भी हो गया. Yes. ये भी हो गया. Next. ये भी हो गया. Yes. Next करते जाइए. मेरे ख्याल से कुछ आ गया इसमें. नहीं आप back. हाँ ये yes. ये बताना जरूरी है. I'll give you one more example here. Dr. Loknath was asking for agro-waste material, agro-waste management. No? Here, you see how we have used the bagasse material. The bagasse we have applied in the shrimp farming in the pond, and the bagasse was very good substrate for beneficial bacteria present in the shrimp farming pond. And you see the biofilm formation, and they were removing the ammonia. Ammonia is very toxic to shrimp. Okay, so it's a kind of plant-assisted bioremediation. We have successfully done it. So agro-waste material can very well be used for even for plant-assisted bioremediation, right? But here, even you can develop a product of bagasse, sell it. You can do that. And bagasse is readily available in the market. आप बाहर जाते हैं किसी भी जूस वेंडिंग मशीन वो जूस निकालता है फ्री में आपको बोलेगा ले जाओ बेगास, हम तो वहीं से लेके आते थे, हमने प्रोडक्ट डेवलप किया वहीं से। तो वही है। नेक्स्ट प्लीज। Okay, so these are the like nanotechnology again, where you can add the nanoparticle in the fish feed, apply, and then you can mix with the zeolite material, apply whichever way you want, and in that way you can mitigate abiotic and biotic stress in the crop plant, animal, and fish. Okay, so review paper we have, you can go through it. Next, please. Then again, here you can see there are many papers and then technology which we have developed, not only with silver nanoparticle, zinc nanoparticle, selenium nanoparticle. See the zone of inhibition here, a nanoparticle size you can see by electron microscope. Okay, and we have used it for fish. Okay, and then we did the micro encapsulation also, filed the patent. So many things we have done it related to nanotechnology. Next, please. So again, the same thing only. This is the paper for biosensor. You can go through it. This is the more elaborate way we have given here related to biosensor about abiotic biotic stress for developing the biosensor. Next, please. Yes, this is very important. Maybe I will finish it within another 5 minutes. Then if you are, because I know many, they have a lot of queries, they are raising their hand, so I will be able to answer it. Here, say Loknath has developed a technology. Now, Loknath wants to develop, he has developed a technology, and Dr. Loknath wants to sell this technology to Dr. Krishnani, to me. Okay. I will ask Dr. Loknath whether do you have any protection of your technology? Okay. Then he will say, yes, I have filed a patent, so it's protected. So I will appreciate him, then I will buy a technology from him. Why? Any technology, entrepreneurship development, any innovative idea, if you have, immediately go for patent. Okay. I'll give you one example. When I was in Maharashtra, Baramati, we were working on some polymer. It's a polysaccharide, hydrogel-based polymer, which was produced by a bacteria. And similar work was being done in neighboring institute in CSI Institute in Pune. I knew that. And I knew that they may file a patent at any time. And they were also working on that. You know what we did it? Our work was incomplete. I told my scientist, whatever work you have done it, go for provisional patent. He went for that. You file a provisional patent. You have 1 year time to file the complete patent. Within 1 year time, we completed the research, and then we submitted the complete specification. Provisional patent, you need only 1 page write-up, only 1 page abstract. Declare it. Okay, that's what he did it. And within 1 year, we submitted complete specification, and then that way we got the priority. Okay, because here 1 second it matters here. 1 second filing. Okay, any idea if you have, you can immediately go for the patent. And here maybe trade secret. Recently we have filed on trade secret also, trademark, and then copyright, and then mostly for the books it is given trademark. You know very well. Okay, trade secret mostly the beverages company like Coca-Cola, Pepsi, they have the trade secret, but trade secret has no protection. By suppose if you come to know what is the formulation of the particular beverages, then no one can sue you because it's a trade secret. It has no complete protection, but patent has the complete protection, and that will be given to you up to 20 years, maximum 20 years. After 20 years, the patent will be in public domain. Anyone can copy after 20 years. Okay, and if you file a patent in India, if you are not filed in the United States, scientists sitting in the United States can very well copy your patent. So that is the reason you have to go for PCT. We file a patent in all over the world so that other cannot copy it. Okay. Then provisional patent, I told you the complete patent. I told you the this one trademark, trade secret, copyright. Okay. And then I think copyright is given up to 50, 60 years, like Kishore Kumar singer. Okay. So and then there is one more here. National Biodiversity Authority in Chennai. If you want to file a patent, anything, whatever biological material, if you used it in the patent application, you need approval from them. Simply 500 rupees you have to pay, simple application, file it, you will get the approval, and then include in the patent application, your patent will be granted if it is based on the novel and innovative idea. Okay, and once it is done, maybe after 18 months, it will be published in the patent journal. Then you have to pay the examination fee. Then if you are from a university, you have to pay only hardly 1,750 rupees. Oh, sorry, 1,750 only. Otherwise, you have to pay 9,000 rupees. if you are from institute, okay? But if you are from university, then it's a very small amount you have to pay. Examination fee also only 4,000 you have to pay instead of 20,000 rupees. So you can save a lot of money on that. Patent related thing, anything, you can contact me at any time, because we file a lot of patent were granted. We never had any patent attorney. We ourselves acted as a patent attorney. Even we appear in the examination, patent hearing, and then finally the patent were granted. Okay. But one thing I want to tell you, while filing patent, you have to do the prior art search. You have to look into that. I'll give you one example. I was in CSR of Canberra that time. I got one letter from Patent Office, New Delhi. There, sir, your patent will be rejected because the patent has no novelty. Then I told him, okay, give me the first examination report. He gave me. In the report, it was mentioned that it's based on the wheat material, and wheat, the गेहूं जो होता है, तो वो उसका already they have done the work in Germany, and in the German language they have published abstracts from 20 years back, whether there's this wheat flour जो आटा है, can remove some of the heavy metal. Then I said, Baba, this is not wheat flour, this is a wheat agro waste material I have used it, which is chemically modified. I simply wrote to them and the patent was granted. So that's what, before filing a patent, I did all this prior art search. I knew that. But you see, it was in the German language, probably I would have never seen it. Okay? But luckily it was in the wheat flour. Had it been on wheat agro waste material, my patent would have been rejected then. Okay? So you have to do the complete prior art search. Then only your patent will be granted. Right? Each and everything in all languages, whether German, French, whichever, you have to go through it and then see that. Okay? Whether it's patentable or not. Simple isolating a bacteria, natural bacteria is not patentable. You have to do some human intervention here, microbial product or something, that is patentable. Okay? Patent related to atomic energy, I think not patentable. So there are so many things you can see, go through it. Okay? Diagnostic. Never ever use this word diagnostic. It is non-patentable, I tell you. One of my colleagues, he filed a patent, but he never discussed with me. He filed a patent, and in the title itself, he wrote diagnostic tool. And later on, when I filed it, I told him it's going to be rejected. And then he say, I'll go patent hearing and he'll see that patent will be rewarded. But it was rejected. Because diagnosis word should not be used. You use, say, molecular tool, you can use some analytical devices, anything you can use it. Okay? Suppose if you have invented a train, ठीक है, ट्रेन से हम जाते हैं, जर्नी करते हैं, न्यू ट्रेन मान लो जिसकी स्पीड बहुत ज्यादा है, जो भी है, जैसे वंदे भारत है, जो भी है, उस पर आप पेटेंट डालना चाहते हैं. If you write here, you have invented a train, okay, the patent will be rejected. But if you write something, you invented a device which will carry passenger at say 200 kilometers per hour, that will be granted. Same invention, same invention, it will be granted. That is called your patent novelty. It should be there. Application should be there. And more importantly, non-obviousness. Non-obviousness का मतलब है सब चीज आप शुरू में बता दोगे, भैया मेरी ट्रेन है, डायग्नोस्टिक टूल है, तो पेटेंट ऐसे रिजेक्ट हो जाएगा. आपको बताना उन्हीं चीजों को बताना है, लेकिन आपकी नॉवेल्टी है, डेफिनेटली 100% नॉवेल्टी है. Use device word. Okay, something like that. Next, please. Now you see here, a lot of this websites are there. You can go through it. Prior art search, you can UK, the Europe, and then also like Germany, France, India, USA, Australia. You get a lot of ideas, prior art search. Okay, and then patent. Sometimes we may, because Dr. Sharma told me that to give some questions, so maybe I'll be giving you some questions to answer it maybe tomorrow. So when we are discussing it, so that you should listen carefully. Okay. Whether the patent is positive right or negative right? It's a negative right, because you are preventing others to use it. It's a negative right, right? Okay.
Yes.
And patent is given for 20 years, right? Provisional patent, file it within 1 year, you can submit complete specification. For biological resources, you need approval from National Biodiversity Authority, Chennai. Okay. Bacteria cannot be, patent cannot be granted. Natural, anything natural cannot be granted. Okay. That's what. Okay. Next, please. Maybe one more slide, then we can have a discussion. Then here we file many patents granted and develop technology, molecular tool, immunization matrix. but don't like, you should not think that already we have done a lot of work, nothing, there's no scope here, but that is mostly related to coastal aquaculture. Okay. Your huge scope, lot of natural biodiversity, fresh water, inland saline areas. Okay. Go there, medicinal plant, go to jungle, collect some sample, do some ethnobotany. Definitely you'll get some novel material, which you can definitely go for entrepreneurship. I tell you, one product, it can make a lot of money. Next, please. Yeah, yes, that's what I wanted to tell you that. This is a very important slide here. 50 to 75% of development money is spent on projects that fail. Where? In US, UK. ठीक है? उनको पता है ये project fail, पता नहीं है उनको. Say they take, say 100 project. They know very well that out of 100 projects, 99 projects are going to be fail. Only one project will be successful. ठीक है ना? 45% of failed projects are related to late of inadequate market analysis. That is true. Okay, he has not done the proper market analysis. 5% project failed because science did not work, and rest failed because of management failures. Okay, there are many overlaps and unexpected things will be there which you never expected it. Okay, because we think there's a linear thing where you can do some basic research, you can do some applied research, develop some technology and commercialize. It's not a linear. Your technology development is not linear. I'll give you one more example here. There was one scientist in the United States. He wanted to develop a toilet seat cover which can be used at the airport so that we can avoid the contamination. And that toilet seat cover is biodegradable. After use, it will be degraded immediately. Wonderful idea. Such a wonderful idea. He got the project from United States, US government, and worked on that. And he developed also. His chemistry worked. He was a chemist. He developed a very good plastic material which is highly biodegradable, which can be used in the toilet. But when he did when he evaluated the cost economics, each plastic material was costing $10. Who will spend $10? It's too much. So that miserably failed because of cost economics. So in the beginning itself, you have to do everything here. Next, please. That is very important. Yes, I think this is my last slide here. You have to image the technology. You have to incubate it. You have to demonstrate it, promote it, and sustain it. Any technology which you develop, global myth is it is the innovation is a linear process. No, it's not a linear process. It's a complicated process. Imaging, incubating, demonstrating, promoting, and more importantly, it's sustaining. Many technology after commercialization, they vanish from the market after 2 or 3 years. But if someone says that he has 100% perfect technology, it means he's telling you the lie. No one has 100% perfect technology. Every technology has some side effect. Take the example of antibiotics which we eat, has many side effects. It is not 100% perfect. So we should keep on working on the technology to make this technology sustainable. Even if you develop any product and have some entrepreneurship, start up something, keep on working on that. Do more research on that throughout your life. Then only it will be successful. Okay. So it's a complex process. That's what you have to do that. And this is a model given by Vijay Jolly. I'm sorry, it's courtesy is Vijay Jolly. It's a book published by Vijay Jolly. Next, please. Next. This, I think somewhere over there. I think this I should discuss in my tomorrow's presentation. There's a few slides are there. Quickly, can you move to the slides? Some more slides. Let's see how many more slides I have. Go ahead. Yeah, please go ahead. Move. Next. I think we can have some discussion now. Okay. Maybe these things I can, maybe if you want, maybe tomorrow early morning. Is that okay for you? Or maybe tomorrow forenoon or afternoon. Tomorrow is holiday for me, but I have to travel from Sonamukhi, West Bengal to Ranchi. It will take minimum 6 hours. Okay, by road. So maybe I'll be reaching there maybe by 12 o'clock or something. Maybe after 1 o'clock, I will be able to take the class. But if you want early morning, I can take. Because I have my second presentation also, but I told you, got correct, something happened. So I have to arrange in the proper way. Okay. But usually I don't give presentation unless I'm fully satisfied. Even for this presentation also, I wanted to have in a proper way, in a proper order. Maybe some slides you can see was not in the proper order. Okay. So maybe if you want, maybe tomorrow early morning.
Rupali ma'am, please let me know about the schedule. How can we arrange, sir?
Actually, ma'am,
Next,
tomorrow we have 3 sessions.
Yes.
From 2 o'clock
Yes.
up to 6:30 we have sessions.
Okay.
So how can we manage, ma'am?
Just after 6:30 to 8.
Sir, 6:30 to 8, actually 6:30 to 7:30. One MCQ is organized, scheduled actually. And after that valedictory is there.
After the validity function will be there.
Yes.
So when can we have the second presentation then? Because that is also equally very important. But tonight I'll prepare it. Maybe it will take
And
me.
can be before 2 if possible, but there will be very hectic. 4 sessions will be there.
I don't know what time I'll be reaching there. So I'll be traveling by road. Okay. My office vehicle is there, car. So maybe in the car I cannot take. I cannot make any presentation. So there'll be internet issue. So, but early morning I can take tomorrow if you want someone if they are interested. जाइए सवेरे 4 बजे उठ जाइए, चाय में पी के तैयार हो जाइए, हम लोग भी बैठते हैं 4 बजे आ जाइए आप लोग। क्या
Is it possible 10 o'clock or
देन पार्टिसिपेंट से पूछिए क्या बोलते हो, और कुछ क्वेश्चंस भी होंगे उनके, पूछिए क्वेश्चंस अगर
Participants, any question you want to ask? It's open for discussion. Now you can ask question by unmuting yourself.
anyway
If
raising
any-
hands.
Any questions and your view about time when the second session to be organized?
Hello, sir.
Yes, yes.
Madam.
Yes.
Yes,
सर
ma'am.
का लेक्चर आफ्टर एमसीक्यू रख सकते हैं मैडम?
After MCQ.
हाँ,
Means 50.
एमसीक्यू का टाइम कम कर दीजिए और सर का लेक्चर रख दीजिए.
But ma'am,
Okay.
valedictory is there, na? So
आप कितने क्वेश्चन दे रहे हैं मैडम?
I'm about 5 questions from each session. So about
That-
60 questions, 60 to 65 questions will be there.
तो
Because
1
agree-
आवर लगेगा, तो थोड़ा पहले शुरू कर दीजिए सेशन मैडम. 5:30 पे डाल दीजिए आप एमसीक्यू एंड देन सर का सेशन 6:30 से रख लीजिए.
maybe I'll finish it within 1 hour. मैं थोड़ा एग्री मार्केट रिवोल्यूशन के बारे में बताना चाहता हूँ, इसलिए मैं थोड़ा सा कोई नई चीज बताना चाहूंगा,
और
इसलिए
सर पेटेंट के बारे में एक क्वेश्चन पूछना था सर.
बोलिए।
पेटेंट फाइलिंग एंड पेटेंट अवार्ड, इसके बीच में बहुत ज्यादा टाइम डिफरेंस रहता है सर. तो कुछ ऐसा है क्या कि फास्ट ट्रैक पे भी होता हो?
यस, एक्चुअली इट्स अ प्रीटी गुड क्वेश्चन. इसमें यह है कि हमने जैसे मैं अपना एग्जांपल दे रहा हूँ, 12 पैटेंट हमने फाइल किए, 7 ग्रांट हुए हैं. जो हमारे पैटेंट ग्रांट हुए हैं मिनिमम 5 ईयर से 7 ईयर 8 ईयर्स तक लगे हैं, हमने 8 ईयर्स तक ग्रांट होने में. आप क्या करिए, द डे यू वांट टू फाइल अ पैटेंट यू गो फॉर कंप्लीट स्पेसिफिकेशन, कंप्लीट पैटेंट. ओके, प्रोविजनल पैटेंट फाइल मत करिए. अगर प्रोविजनल करते भी है तो विद इन 1 ईयर आपके पास टाइम है सबमिट करने का. एंड देन यू पे द एग्जामिनेशन फी, गो फॉर द स्पीडी पैटेंट. स्पीडी पैटेंट में आपको पे करना पड़ेगा उसमें कुछ. ओके
जी.
बट इट विल नॉट बी, it will be after 18 months only, because after 18 months only it will be published in the patent journal abstract. After that only your patent will be granted.
Sir, for design patent,
Anything design or chemical or microbiology or biotechnology or physics,
okay sir.
electronics department are there, biotechnology भी है, तो उसमें सब में मिनिमम उनका 18 months के बाद ही आपको देंगे. लेकिन you go for speedy patent वो पे कर सकते हैं आप, एक्स्ट्रा पे करिए उसमें उनको कि आई वांट टू गो फॉर एग्जामिनेशन इमीडिएटली, ऐसा कर सकते हैं.
क्षमा मैडम, सब आपके लिए था ये क्वेश्चन.
मैम
एक्चुअली एग्जामिनेशन का जो है ना, आप
हाँ।
उसमें उनको क्या बोलते हैं, फी पे कर दीजिए, एग्जामिनेशन फी, और साथ में पेटेंट हियरिंग होगा। लेकिन आजकल थोड़ा जल्दी हो रहा है क्योंकि ऑनलाइन हो गया आजकल। पहले हमारे समय में ऑनलाइन नहीं था, पूरा फिजिकल मोड पे होता था, हार्ड कॉपी
तो
सबमिट
फिर भी 3 to 4 months मिनिमम लगता है। और किसी किसी केस में जब FER में कुछ प्रॉब्लम, जैसे आप बताए फर्स्ट रिपोर्ट में कुछ अगर प्रॉब्लम आ जाता है तो वो थोड़ा सा टाइम और लगता है सर।
लगेगा। दूसरा,
months-
मेरे 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 पेटेंट का डिले मैं आपको बता रहा हूँ कहाँ पे हुआ। मुझे पता ही नहीं था कि मुझे बायोलॉजिकल सोर्सेज का अप्रूवल लेना है नेशनल बायोडाइवर्सिटी अथॉरिटी से, क्योंकि मैंने आपको बताया कि मैंने कभी भी पेटेंट अटॉर्नी को हायर नहीं किया।
ओके।
मैंने खुद ही, क्योंकि एक पेटेंट जहाँ हमें मात्र 10 या 20,000 रुपीज खर्च करने हैं, या मैक्सिमम 50,000 रुपीज टिल ग्रांट, 1 लाख मान के चलिए, उसी पेटेंट को ग्रांट कराने में वो पेटेंट अटॉर्नी 2 लाख रुपए लेता है। मैंने कहा नहीं, मैं सीख के खुद ही करूंगा, खुद सीखा, हमने एक पैसा नहीं दिया उनको, खुद ही किया सब कुछ। तो उसमें एक कमी ये रह गई कि नेशनल बायोडाइवर्सिटी अथॉरिटी को हमें मिस हो गया था। तो बाद में वो पेटेंट कंट्रोल ऑफ एग्जामिनेशन ने मुझे एडवाइस किया कि सर आप जाइए NBA ऑफिस में अप्लाई करिए, अप्रूवल लेके आइए, हम आपका पेटेंट ग्रांट कर दूंगा। फिर हमने किया, उसी वजह से 1 साल डिले हुआ। तो पहले होता तो हम पहले अप्लाई कर देते। तो NBA को तो आपको अप्लाई करना ही है, तो आप वो टाइम आप थोड़ा सा यू कैन ब्रिज द गैप, वो आप कर सकते हैं।
सर कल के लिए एक काम करते हैं, सबके प्रेजेंटेशन में से थोड़ा थोड़ा टाइम कम कर लेते हैं और 1 hour हम निकालते हैं, है ना, आपके लिए। एंड देन वी विल बी टेलिंग यू आफ्टर 5 यू विल बी एबल टू प्रेजेंट।
यस डेफिनेटली, उनसे पूछिए, मैं तो अवेलेबल हूँ।
हाँ, और बाकी तो हम लोग तो 2 बजे से अवेलेबल हैं सर। तो वो क्या है, थोड़ा थोड़ा सा टाइम और थोड़ा पहले चालू करें। और प्रिंसिपल मैम का कहना है कि वी हैव टू टेक केयर ऑफ पार्टिसिपेंट्स डाटा आल्सो एंड मोबाइल चार्जिंग आल्सो। So we will be giving 10 minutes break for that, so that they can get their mobile or any other accessories be charged. And data charge, so the participants, there's a request that please have some extra charge. If you are taking Airtel or Jio, it will be costing 19 rupees or 22 rupees for a full day. If extra charge is required, your data gets finishes. So you can have that much for a very good session as tomorrow's session. And we will be having 4 sessions tomorrow, and your exam also. Along with that, a valedictory session of just half an hour, I think. So our valedictory session will not be a huge one. It will be on online platform only. And so if someone wants to ask any question, please.
Or if you give me 1 hour time,
Yes, sir.
maybe it was 9, okay, 8:45, no? Maybe 10:30 I can take.
No, sir, it will not be possible for us. Today, 10:30, we might be like that myself and Rupali ma'am will be joining, and others, because, yes, Loknath sir, please ask your question. You have raised hand.
Yes.
Dr.
Hello, sir.
Loknath Deshmukh, please.
Yes.
Hi, sir.
Boliye.
Thank you so much, sir. And obviously, sir, most of us will love to listen you with no time limit. But according to the management, ma'am is fixing the things. And my question is that how much they are charging for, sir, or that National Biodiversity Authority, and they are charging for each and every experiment, or what sort of?
No. I'll tell you. I'll give you one example, then it will be very clear to you. My patent was on molecular tool to examine the microbial diversity in coastal aquaculture. Okay, in coastal soil. Okay, I repeat, in the coastal soil to examine the microbial diversity, bacterial diversity using metagenomics. Okay, that was a molecular tool. So my patent was about to be granted. Everything was clear because it was 100% novel. The primer which I developed, 100% novel. Everything was very clear. PCR, whatever. Okay. But my patent officer told me we need approval from NBA because you have used the soil. Then I argue with them. I argue with them. I said, I have used the soil. Why should I go for that? Is it a biological material? I knew very well it is a biological material because I myself was contradicting it. Because you see, I am using them
Yes, sir.
metagenomics, bacterial diversity in soil. Okay. Then I had no option but to go to the NBA. Soil, just for using soil. You know how much soil? 0.5 gram only. We used a lot of replica. Lot of replica we have used it. But that soil also different places we collected. Nagapatnam, Tamil Nadu. So that they gave me the approval. I paid only 500 rupees that time. So only 500 rupees we have to pay. So they will give the approval, then you can apply and then patent will be granted. Even the post approval also you can get it. No issue. Post means like once you file the patent, later on you can get the approval from NBA. Yeah. Any more?
Any more questions?
Thank you, sir.
Yeah. Well, when I was presenting, many participants were raising their hands. Now they are silent. What happened?
It might be, they might be waving waving for you, and the button for raising hand was there.
Yes, by mistake, something.
So by mistake.
ठीक, कोई बात नहीं, कोई issue नहीं.
So,
Okay.
once again, sir, your thought-provoking and academically rigorous lecture on climate smart innovations in agri market revolution has been truly invaluable. You not only highlighted the emerging climatic smart agricultural technologies, but also eloquently connected them with the digital supply chains, blockchain and traceability, e-market platforms, value-added processing. And your insights provided our participants with a much-needed global perspective on how agricultural markets are being reshaped in the face of climate change and technology disruption. The way you contextualized self-employment, innovation-driven startups, and rural economic empowerment has left a lasting impression on all participants. Your academic depth, combined with pragmatic illustrations, bridge the gap between theory and practice, showing how research translational technology commercialization and entrepreneurship can become pillars of sustainable development, moreover, agricultural development. Your lecture has not only broadened our participants' academic, ignited in them a spirit of innovation, entrepreneurship, and climate resilience. The clarity with which you addressed the queries during the interaction session further enriched the learning experience, reflecting your generosity as a teacher and as a mentor. On behalf of the organizing committee, the management, the principal, and all the participants who have joined us from across India, I extend our sincere thanks to you, and your guidance has added immense value to this FDP and will certainly inspire our participants to pursue impactful research, innovations, and entrepreneurial ventures in the agri-food sector. We look forward to continue academic association with you and to draw inspiration from you, exemplary contribution to agricultural sciences and innovation. Thank you once again, sir, for gracing us and taking out time from your busy schedule while moving from your place and attending a meeting at Calcutta, West Bengal, and then after a hectic schedule, presenting a presentation of more than 3 hours. Thank you so much, sir. And Rupali ma'am, please continue for formal vote of thanks.
Okay, thank you, ma'am.
So finally, what has been decided? Tomorrow, what time? 1 hour?
After second half, sir.
Okay, I'll be available. Just 1 hour, maybe even 45 minutes. My
Okay.
slides will be ready. Okay.
Okay, sir.
And I can share those slides with the participant also.
Okay, sir. Okay, sir.
Okay, sir. With this, we conclude this day of our FDP. On behalf of organizing team, I sincerely thank our expert, Dr. Kishore Kumar Krishnani sir, for delivering such an extremely rich and thought-provoking session today. I also thank our coordinator, Dr. Shamai Beg ma'am, and all the participants for their active involvement. Now I would like to invite Ms. Yogita Lokhande to deliver the vote of thanks, formally deliver the vote of thanks. Please, Yogita.
Thank you so much, ma'am. As we bring day 5 of the AICTE ATAL FDP on technological innovation in agrotech and food processing to a close, it's my privilege to extend a warm vote of thanks on behalf of Swami Sri Swarupanand Saraswati Mahavidyalaya. We are truly honored to have with us Dr. Kishore Kumar Krishnani sir for delivering thought-provoking session on climate smart innovation in agri market revolution. Dr. Krishnani emphasized the planning and steps involved in establishing business, along with details of funding sources such as venture capital, crowdfunding, loans. You also highlight the concept of nutritional biotechnology, integrated bioremediation system, biosensor, and crucial importance of intellectual property. His guidance truly served as a roadmap for empowering students and participants to transform their idea into impactful enterprise. Thank you so much, sir. We remain deeply grateful to our patrons whose vision and encouragement have been the constant strength behind this FDP. I would also like to acknowledge the leadership of our principal, Dr. Hansa Shukla ma'am, whose unwavering support and motivation continue to inspire us. Special appreciation is due to the faculty coordinator, Dr. Shama E. Beg ma'am, and co-coordinator, Mrs. Rupali Kharche ma'am, and whole organizing team whose tireless effort and teamwork ensured the seamless execution of today's program. Finally, my heartfelt thanks go to all the participants whose enthusiasm, engagement, reflective questions, and active involvement added immense value to today's session. Before concluding, may I kindly remind everyone to fill out the feedback form of today's session and join tomorrow's session on time for another enriching day of learning. Thank you so much.
Okay.
Thank you,
Thank
sir.
you. See you then. See you then.
Yugal Kishore sir has a message for you, sir. That Professor KKK, fortunate to hear you again on online mode. Yugal Kishore sir, whom you met at that international conference at our place
Yes, yes.
from Meghalaya.
Okay,
Yes.
achha.
Yes.
Okay, right.
Yes.
Thank you. Okay, then. Thank you all the way. See you then.
Thank
See you
you, sir.
tomorrow. See you
Good
tomorrow.
night,
Okay.
sir. Good night, sir.
Okay, see you then. Yeah.
Okay.
Take care. Good night. Bye-bye. Yeah.
Bye, sir. Good night to everyone.
Good night, ma'am.
Good night, ma'am.
Good night, ma'am.